explore the fascinating journey of human evolution, tracing the lineage from our common ancestors with the great apes to the development of unique human traits such as articulate speech, abstract reasoning, and complex social structures. This episode examines the latest scientific discoveries that illuminate our genetic connections with other primates and how these findings enhance our understanding of ourselves. The journey begins with the groundbreaking work of the telomere to telomere (T2T) consortium, which has achieved a monumental milestone by piecing together the complete genetic blueprints of the sex chromosomes of several great ape species, including chimpanzees, bonobos, gorillas, Bornean and Sumatran orangutans, and the siamang gibbon. This research, documented in the journal Nature, provides new insights into the dynamic evolution of the Y chromosome, shedding light on health, reproduction, and evolutionary trajectories shared between apes and humans. Through meticulous analysis, researchers have discovered that while over 90% of the ape X chromosome sequences match human sequences, the Y chromosomes show a faster rate of evolution. Interestingly, the Sumatran orangutan's Y chromosome is notably longer than that of the gibbon, primarily due to the presence of repetitive DNA, which plays a critical role in functions like sperm production. These genetic sequences do more than fill gaps in our understanding of ape and human evolution. They also offer crucial data for conservation efforts. As many great ape species face threats of extinction, understanding their genetic makeup is essential for developing effective conservation strategies. Moreover, the insights gained extend into the realm of human societal perceptions, particularly the misconceptions surrounding the Y chromosome, historically stigmatized and linked with aggression and criminality. Modern genomic research debunks these myths, reinforcing that DNA does not dictate complex social behaviors. This narrative of genetic exploration not only revisits our evolutionary past, but also opens new doors for future research in comparative genomics. It paves the way for further studies that could unravel more about what distinctly makes us human, beyond just our genetic makeup. By delving deeper into the genetic fabric shared with our closest living relatives, scientists continue to piece together the puzzle of human evolution, enhancing not just our knowledge of biology, but also informing approaches to preserve the biodiversity of our planet. This ongoing quest underscores a broader scientific endeavor to safeguard our shared genetic heritage and the myriad life forms on Earth. Continuing from the exploration of our genetic ties with the great apes, let's delve deeper into the specifics of the groundbreaking research on the X and Y chromosomes, which are pivotal in understanding both the similarities and the stark differences that define our species within the tapestry of evolution. This segment focuses on the intricate details of how chromosome sequencing has been instrumental in mapping the evolutionary lineage of humans compared to our closest primate relatives. The research undertaken by the telomere to telomere T2T consortium marks a significant advancement in genomic studies. The consortium's efforts to decode the complete genetic blueprints of the sex chromosomes across multiple great ape species have unveiled critical insights. The sequencing of the X and Y chromosomes has not only filled substantial gaps left by previous genomic projects, but has also clarified the evolutionary paths that diverged and led to the distinct biological identities of each species. One of the most striking findings from this research is the high degree of similarity in the X chromosomes across species. Over 90% of the sequences in the ape X chromosomes align closely with those found in humans, suggesting a strong genetic linkage and a relatively recent common ancestry. This similarity underscores the subtle yet profound genetic shifts that have occurred over millions of years, leading to the diverse array of great apes and humans seen today. In contrast, the Y chromosome tells a story of rapid evolutionary change. The Y chromosomes exhibit far less similarity across species, indicative of a faster rate of genetic drift and adaptation. This rapid evolution of the Y chromosome could be linked to its roles in male fertility and reproduction, areas that are often subject to intense selective pressures. The differences in the Y chromosomes among species are so pronounced that they provide a clear genetic marker of the evolutionary trajectories that have led to the uniqueness of each species. Moreover, The variability in the length of the Y chromosome among different species, as highlighted by the significant difference between the Sumatran orangutan and the gibbon, points to the dynamic nature of genetic evolution. These differences are largely due to the accumulation of repetitive DNA sequences, which can play crucial roles in gene regulation and expression. Such findings are vital for understanding the genetic basis of many species-specific traits that influence reproduction and survival. The detailed sequencing of these chromosomes not only informs us about our past, but also has direct implications for the present and future, particularly in the fields of medicine and conservation. By understanding the genetic foundations of our closest evolutionary relatives, researchers can better predict certain genetic diseases, understand genetic disorders, and develop more targeted treatments. As this research continues to unfold, it becomes increasingly clear that the X and Y chromosomes are more than just carriers of genetic material. They are historical documents, inscribed with the codes of evolutionary change, that when thoroughly examined, reveal the interconnected stories of life forms that share this planet. Through such scientific endeavors, the shared genetic heritage and the divergent paths that have shaped the great apes and humans are brought to light, offering invaluable insights into the complexity of life and evolution. Building upon the deep genetic insights gleaned from the sequencing of the X and Y chromosomes, this segment shifts focus to the evolutionary timeline, particularly the significant divergences that mark the journey from our closest primate relatives to the emergence of Homo sapiens. This exploration delves into how specific genetic mutations and environmental factors have carved out the distinct traits associated with modern humans. The evolutionary split from our primate cousins is a narrative etched deep in our genetic codes. Scientific investigations reveal that the lineage leading to modern humans diverged from that of chimpanzees and bonobos, our closest living relatives, around 6 to 7 million years ago. This pivotal divergence set the stage for the unique evolutionary pathway that would eventually lead to the rise of Homo sapiens. As the human lineage branched away, several key genetic mutations played crucial roles in shaping what would become distinctly human traits. One of the most profound changes was in the gene FOXP2, often referred to as the language gene, which underwent significant changes only in the human lineage. This mutation is closely associated with the development of complex language skills, setting the foundation for articulate speech, a hallmark of human communication. Another crucial genetic shift involved the brain's development, particularly the expansion and increased complexity of the neocortex, which plays a significant role in higher-order functions such as reasoning, planning, and socializing. This expansion didn't occur in isolation. It was influenced by a myriad of environmental factors. The shift from dense forests to more open savannas, for instance, presented new challenges and opportunities that favored bipedalism and tool use, traits that would become prominently developed in early humans. Environmental pressures and changes also played a pivotal role in shaping human evolution. Fluctuating climates, for instance, likely drove the need for adaptive strategies that led to the development of technology and controlled use of fire. These adaptations not only allowed our ancestors to survive diverse and changing landscapes, but also led to changes in diet and social structures, further influencing genetic evolution. The development of cooking and the control of fire also had profound implications for human evolution. The ability to cook food fundamentally changed the nutrient profile of diets, allowing early humans to feed more energy-demanding brains. This dietary change is reflected in the genetic adaptations around metabolism and digestion found uniquely in humans. As these genetic and environmental factors intertwined, they propelled the evolution of early hominins through various species such as Homo habilis and Homo erectus, each marking progressive steps towards the anatomically modern Homo sapiens. This journey was not linear, but a complex web of evolutionary branches, many of which did not survive the passage of time. Today, the tools of genomics and archaeology are unraveling this intricate history, highlighting the interplay of genetics and environment in the evolutionary saga of humans. Each discovery adds a piece to the puzzle, helping to trace the lineage and shed light on the myriad factors that contributed to the emergence of the species known today as Homo sapiens. Through this lens, the story of human evolution is seen not just as a series of genetic accidents, but as a dynamic and ongoing interaction between biology and environment. Moving beyond the realm of genetics and evolutionary biology, this segment explores the profound implications of recent genetic findings on our understanding of human behavior, social structures, and cultural developments. By examining how our genetic makeup interacts with environmental and social factors, we can gain deeper insights into the complexities of human psychology and society. The genetic underpinnings discovered through the study of the X and Y chromosomes and other genetic markers do not just narrate the story of human evolution. They also provide a framework for understanding how certain behaviors and societal structures may have developed. For instance, the genetic adaptations related to the FOXP2 gene, which is associated with language and communication, underscore the importance of social interaction and collaboration in human survival and success. This gene's evolution suggests that the ability to communicate effectively played a crucial role in the formation of complex social structures that are uniquely human. Moreover, genetic variations that influence serotonin and dopamine, neurotransmitters involved in mood regulation and reward pathways, offer insights into the biological foundations of human emotions and social behavior. Understanding these variations can help explain the diversity in human psychological experiences and the varying susceptibilities to mental health disorders across different populations. The integration of genetic research with studies in anthropology and psychology also enhances our understanding of cultural developments. Cultural practices, from dietary habits to ritualistic traditions, can be seen as adaptations to both environmental conditions and genetic predispositions. For example, the development of lactose tolerance in certain populations illustrates a clear interplay between a genetic mutation and a cultural shift towards dairy farming, which in turn influenced social and economic structures. Furthermore, genetic research challenges and refines the concept of what it means to be human. By highlighting the shared genetic heritage with other primates and tracing the unique mutations that led to human-specific traits, genetics fosters a broader perspective on human identity. It prompts a reevaluation of the boundaries between biological determinism and the social constructs that define human society. Additionally, these genetic insights provoke ethical considerations about the manipulation of human genetics and its implications for future generations. As science advances, the potential to alter genetic destinies through technologies like CRISPR raises profound questions about the nature of human evolution and the values that society cherishes. In conclusion, the implications of genetic research extend well beyond the confines of biology, influencing theories in psychology, sociology, and ethics. As we continue to unravel the genetic threads that weave the tapestry of human life, it becomes increasingly apparent that our genes not only contribute to our physical form, but also shape the essence of our cultural and social identity. This holistic view encourages a deeper appreciation of the intricate interplay between our genetic makeup and the environments in which we develop, live, and interact. As we deepen our understanding of genetic heritage and its implications for both humans and our closest evolutionary relatives, the great apes, it becomes imperative to address the intertwined issues of conservation and the ethical dimensions of genetic research. This segment explores how the insights gained from genetic studies influence conservation strategies for great ape species and the ethical considerations that these scientific pursuits entail. The close genetic ties between humans and great apes underscore a shared evolutionary history and present compelling reasons for the conservation of these species, many of which are endangered due to habitat destruction, hunting, and other human activities. Genetic research has not only highlighted the biological and behavioral similarities we share with these primates, but has also provided vital data that can be used to enhance conservation efforts. For instance, understanding the genetic diversity within populations of great apes enables conservationists to manage breeding programs more effectively and helps in mapping out conservation strategies that can maintain or enhance this genetic diversity. Moreover, the the sequencing of the great apes' genomes offers insights into their health susceptibilities, which are similar to those of humans, such as susceptibility to certain infectious diseases. This knowledge is crucial for developing better medical treatments that can be administered in the wild or in sanctuaries, thus improving the chances of survival and reproduction for these species. On the ethical front, the use of genetic research raises several questions. One primary concern is the extent to which humans should intervene in the natural processes and lives of other species. While genetic tools can be used to conserve species, they can also be employed in ways that alter them through genetic modification. The possibility of cloning or genetically modifying great apes, for example, presents a controversial scenario that could challenge the natural evolutionary processes and potentially lead to unforeseen consequences. Furthermore, as we uncover more about our genetic similarities with great apes, the ethical considerations of how these animals are treated, both in the wild and in captivity, become more pressing. The realization that great apes possess complex emotions and social structures similar to humans challenges existing perceptions and treatment of these beings, calling for a reevaluation of their rights and the protection they are afforded by law. Another ethical consideration involves the consent and privacy issues related to genetic research. While humans can provide consent for genetic studies, the same is not true for great apes, raising questions about the exploitation of these animals for scientific gain. Additionally, as genetic data can be sensitive and potentially misused, establishing stringent regulations to govern who can access and use this data is essential to protect both human and non-human subjects involved in such research. In conclusion, understanding our genetic heritage not only enriches our knowledge of human evolution, but also brings to light our responsibilities toward other species with whom we share this planet. It compels us to consider more deeply the ethical frameworks within which genetic research operates and the broader implications of our conservation efforts. As we navigate these complex issues, it is crucial that ethical considerations guide our actions, ensuring respect and care for the great diversity of life that enriches our shared ecological and evolutionary heritage.